Get started. I am standing in for Matt today. Thanks to everyone for coming to I think it's the first of the semester. So thank you, Stephanie, for being willing to kick off our seminar series. I'm personally really looking forward to this one. Um, just to give a little bit of background, Dr. Stancil is an adolescent medicine clinician scientist. Um, I'm sorry, my dog is not happy with me right now. In the divisions of adolescent medicine and clinical pharmacology, toxicology, and therapeutic innovation at Children's Mercy Hospital, and an assistant professor in the Department of Pediatrics at the University of Missouri, Kansas City School of Medicine. So you keep yourself very busy. Her clinical practice has focused on complex teens in specialized and vulnerable populations for over a decade. And her interdisciplinary translational research focuses on developing precision therapeutics for adolescent mental health conditions by understanding variability in exposure and response. So Dr. Stancil, thank you so much for joining us. And everyone, if you will do questions toward the end, and you're welcome to put them in the chat, and Cassie will be monitoring that, or you can ask via camera. So go ahead, Stephanie.
Great, thank you. Thank you all for inviting me to be here this afternoon. I am hopeful that even though we're not talking about autism specifically today, that the approach to kind of applying this precision therapeutics inquiry and to answer some clinical questions might be of use to those of you who have your own kind of clinical questions that you'd like to answer in the research setting. All right, so we're going to focus today on eating disorders, and so I'd like to provide a bit of background about that. Eating disorders are fairly common in teens. They affect up to 5% of youth and include things like anorexia nervosa, bulimia nervosa, and binge eating disorder. They can affect teens of all genders, races, ethnicities. Essentially, you can't look at someone and tell whether or not they have an eating disorder. They are associated with high morbidity, things like cardiac compromise, malnutrition, substance use disorder, and mortality. Anorexia nervosa has the second highest mortality rate of any psychiatric condition. It used to be the first, but was usurped following the opioid epidemic. And importantly, there are no medications that are approved for treatment of eating disorders in pediatrics. There are a couple available that have been approved in adults, like fluoxetine for bulimia and Vyvanse for binge eating disorder. But again, nothing has been approved in pediatrics, and so all medications used are off-label. We know that there are reward system alterations in eating disorders, such that there appears to be an increased taste perception, perhaps a lower kind of pleasure threshold, and potential kind of altered inhibitory controls in patients with eating disorders. Some folks with eating disorders have impulsive or compulsive behaviors, like the binge eating specifically, or purging, which can be vomiting after eating, compulsive exercise, use of laxatives, these types of purging behaviors to prevent weight gain and rid the body of nutrition. And these behaviors seem to activate the reward system. So enter naltrexone. Naltrexone is a medication that binds and blocks the opioid receptors. It's FDA approved for alcohol and opioid use disorder in adults and used across the lifespan for a variety of conditions. Specifically in pediatrics, it's used for things like non-suicidal self-injury or self-harm. It's used for impulsivity and autism. It's been used in Prader-Willi, among other things. And if you look across all of these different indications, response varies, usually between 40 and 80%, depending on the study. Specifically for eating disorders, naltrexone has been studied in adults, but the findings are mixed. And the optimal dose for treatment of eating disorders is really not clear. So some studies support that you need doses, up to 400 milligrams a day to get reduction in binge eating or purge behaviors. And that's 8-fold higher than the FDA-approved dose, that's shown to be effective for opioid use disorder, for example. So then you wonder, well, are patients with eating disorders truly processing this medication differently, such that they need higher doses? Or is the kind of mechanism of disease different? Is the target different? Are we treating something else? And so you have this table over here on the bottom right-hand side. And naltrexone not only binds and blocks the mu opioid receptor, which is kind of the most famous opioid receptor, if you will, and associated with euphoria, but also can activate or rather bind and block kappa and delta opioid receptors as well, although with decreasing affinity. So if you have a higher dose, if you're truly getting higher exposure in the brain, that begs the question, are you actually blocking kappa and potentially delta and getting those effects muted as opposed to simply mu? But we don't know. So as a clinical pharmacologist, these types of questions are interesting to me, and I've been trained to think about them kind of under this classic paradigm of clinical pharmacology, where we think of a dose that gets prescribed for a patient. That dose of a medication reaches a certain exposure in the body, meaning that the amount of drug available for use in the body can be measured. And then ideally that exposure leads to a response. We want our patients to feel better quickly, and ideally kind of the first time with that first medication. And so we here now have this kind of drug that's used for a variety of things. It seems to work somewhat in adults. And it happened to be used in our regional eating disorder center at Children's Mercy as well for teenagers. But how effective is it, and how much should patients be getting? We didn't really know. So that's what set us off on this quest. It actually started during my fellowship. And I talked with my colleagues and kind of identified this drug and this kind of clinical conundrum as something that had a lot of unknown, and so a lot of potential to learn and to attack it from different angles. And so we'll talk about some of that today. So the first question that we wanted to ask is, how specifically is naltrexone being used in teens with eating disorders? Again, there was some data in adults with eating disorders. It was all pretty old, from the 80s, early 90s. And again, we were using it in our eating disorder center, but how specifically it was being used and then what the response was like, we didn't know. So we started with a retrospective case series, and we looked at all the patients who had been prescribed naltrexone as part of their clinical care. And what we found is that the majority, about 2/3, seemed to have reduction or elimination of their binge or purge behaviors if they were treated with naltrexone. Now, it's important to note that this is in the milieu of kind of this multidisciplinary treatment environment. So they're not only getting medication, they're getting family therapy, they're getting individual therapy, nutrition services, etc. But regardless, based on this kind of retrospective case series, 2/3 responded. Seemed to be safe and well tolerated. We looked for safety signals with liver enzymes and a few other things, and found no association with kind of serious adverse events in naltrexone use. And I pulled this quote from this particular study because I think it's really illustrative of what we think is happening when a patient is treated with naltrexone for these behaviors. She continues to purge, but less frequent. Last episode was weeks ago, but couldn't remember the exact date. She says she feels awful afterward. It no longer gives her the sense of calm. So when we think about what the target truly is, this would make sense if we were, binding and blocking the opioid or the mu opioid receptor that would block that euphoria or block that calm associated with these behaviors. But again, it just wasn't clear in kind of a more granular way. So from this study, we said, okay, great. 2/3 of folks respond, but that's still 1/3 that doesn't. And what's going on with them? Is it related to dose? Is it related to some other factor? Can we figure that out so we can predict who's going to respond sooner? Again, kind of going back to my clinical pharmacology training, this is a paradigm kind of introduced to many of us by Dr. Steve Leder, who some of you may know or work with, and has taught us to kind of systematically approach some of these clinical questions, ticking off these different elements. So for this particular example, we have a drug that works for some patients, but not everybody. And so we can ask ourselves, what pathways are involved in drug disposition. So how the drug is being either absorbed, metabolized, excreted, et cetera. What are the pathways involved in drug response? What are the targets? Is there variability at the actual receptor, for example? We can then also ask ourselves, what is the developmental trajectory of those pathways? So if we can identify how the drug is metabolized, for example, does age impact that? And if so, does that impact how much drug these particular patients see when they're prescribed? Additionally, are there functionally relevant genetic variants that could impact variability? And what is the intersection between genetic variability and the impact of age? And so some of these questions we'll kind of go through today, but this was really the scaffolding for this project early on. So this slide here is the disposition pathway of naltrexone, and I'll kind of guide us through it. Hopefully you all can see this laser pointer. So naltrexone is abbreviated NTX here. So to answer that first question, what are the pathways involved in naltrexone disposition? We know that naltrexone, when administered orally, it is absorbed through passive diffusion from the gut into kind of the portal vein and gets delivered to the liver. The liver kind of readily metabolizes naltrexone to this primary metabolite, 6-beta-naltrexol. And it does so through this family of enzymes, the AKR1C family, and primarily AKR1C4. We're going to talk about this enzyme quite a bit in the next couple of slides, so I wanted to highlight that here. Once 6-beta-naltrexol is formed, it can get further metabolized through glucuronidation, but eventually can get excreted renally, as well as potentially kind of reabsorbed through the GI tract through a process called enterohepatic recirculation. But we'll kind of focus today here on this AKR1C4. So that is one of the pathways, one of the main pathways involved in naltrexone disposition. So then we wanted to ask ourselves, okay, what's the impact of age and genetics on this pathway? I looked into the literature first, and not surprisingly, there wasn't much of anything known about the impact of age. And so we set out to answer that question through some in vitro work, where we utilized a pool, or rather a kind of a cohort, not a pool, but a cohort of individual liver donors that were both pediatric and adult, from 5 different sources across the country. One large source that contributed these individual donors was University of Maryland Biobank that stores liver samples from patients post-autopsy. And then there are other liver donors samples that are obtained through, if there's a liver being prepared for transplant, for example, or within an hour after death, right? So there's different kind of ways that these samples are obtained, which turns out matters, and we'll talk about that in a little bit. But from these 5 different sources, it made up this cohort of 158 liver samples. And we looked at the activity of naltrexone biotransformation into this metabolite, 6-beta-naltrexol, in these individual livers. And what we found is that there was substantial variability from sample to sample, from liver to liver, greater than 200-fold difference in rate from one donor to another. Once we accounted for some of the variability associated with source, that variability went down to about 20-fold, but still substantial, right? So meaning that the ability of those particular livers to metabolize naltrexone varied 20-fold or more in an in vitro environment. We were able to look at the impact of age on naltrexone biotransformation, and we do so by kind of quantifying the formation rate of the metabolite. And what you can see of this figure on the left here, when we look at age kind of as a categorical variable, you can see that there may be a slight increase until about 2 years of age before it plateaus through adulthood. I will say that that's limited by the smaller number of samples we had in kind of the younger than 2 age group. But beyond kind of age itself, if you look within each of these categories, you can see substantial variability, looking at the top to the bottom data point, that is far greater than it is across the age groups. So age maybe matters, but it seems like there's still substantial variability to be accounted for. We also wanted to look at the impact of genetics. There was a little bit of data supporting the potential for this genetic variant in AKR1C4 reducing activity of that enzyme, meaning it would be less active at metabolizing naltrexone or other medications through that pathway. That data that was existing was in recombinant enzymes, so in a non-kind of human system. And so we wanted to look at it in a more intact kind of human, albeit in vitro environment. And so when we looked at donors who were either wild type, so they did not have the genetic variant, compared with those who either had one copy of the variant, on average, the individuals who had one copy of these was actually co-occurring missense mutations, they had on average a 30% reduction in activity of that particular pathway. And then if we look at individuals who had two copies of that genetic variant co-occurring missense mutation, on average, they had a reduction in 71%. So this suggested that indeed this genetic variant seemed to be reducing activity of this pathway, the ability for naltrexone to be metabolized in these donors. We looked at that a little bit further, kind of taking that cohort of 158, but trimming it just to pediatric samples, 0 to 18, which left us with 73 donors. And we adjusted it for source, so we could kind of take that element of variability out and look at the impact of age and the variant through a model predictive of what that activity would be. And you can see in the table here, age in decades had, a moderate effect size. But again, if you think about age in decades, so the first decade of life, the second decade of life, it really in context is not likely that meaningful. Sex, again, not likely that meaningful, but in the blue box here, you see the variant, AKR1C4 co-occurring variants, and that did seem to matter. I mean, the effect size here was pretty substantial. So that was interesting to us. And we thought, okay, from this in vitro work, we've now kind of characterized the impact of age on naltrexone metabolism and also looked at genotype. And from this data, genotype seemed to matter a whole lot more than age did. If we did have to kind of speak to a breakpoint and when biotransformation reached that of adult levels, perhaps around 2 years of age. But most importantly, source seemed to matter with this work, as there was an impact on what we think is kind of protein quality in these samples that come from different sources. Happy to answer questions about that afterward, but I don't want to spend a ton of time on that to get to some of the more clinical stuff. So the next question was, okay, so here we know that these couple of variables might impact naltrexone metabolism, but what does this look like in actual humans, right? So the next question we asked was, what does systemic exposure look like in teens treated with naltrexone for their eating disorder? So this is a conglomerate of 2 different studies where we enrolled patients who they were eligible if they were between 12 and 21, who were on naltrexone as part of their clinical care. And we did essentially a 12-hour PK. The majority of this cohort was female and had a wide range of weight and BMI, which is consistent with the variety of different eating disorders that were kind of seen within this study. You could have anorexia nervosa binge purge subtype, which could be individuals who struggle with substantial restriction of food intake in addition to either binge or purge. And you can also have individuals on the other end of the spectrum with maybe binge eating disorder, whose BMIs are often in the obese or morbidly obese range. And of this 21 adolescents who participate in this study, 7 of them had this co-occurring missense mutation in AKR1C4. And so what you see here on the right-hand side is each individual's kind of concentration time curves for naltrexone. And just looking at it really quickly, it's kind of all over the place, right? I do want to draw our attention to the y-axis, that's log transformed. And so when you think about the top point here compared to one of these other ones down here, that's, several orders of magnitude different, potentially than maybe what it would look like at first glance. So in some of the pharmacologic parameters that are important to us when we think about what's going on with drugs in the body, one is the maximum concentration in plasma. We call that Cmax. And you can kind of visually pick that out for each curve here. Here's the Cmax for this individual. Here's this one for this one. And hard to tell in here, but that's particular parameter is useful to tell us that peak concentration. And when we look at each individual Cmax, there was 63-fold variability from patient to patient. And this was already normalized for milligram per kilogram dose. So once we took kind of dose and weight out of the equation, we still had 63-fold variability in Cmax. Looking at area under the curve down here, that's another pharmacologic parameter that's useful to us to tell us kind of total systemic exposure. So kind of how much drug is available for use in the body over a period of time or over a dosing interval. And you can calculate that essentially by if you look at any one of these curves and you would shade underneath kind of a particular, you shade in that area, you calculate out kind of what that total exposure is. And so looking at area under the curve from individual to individual, again, already dose and weight normalized, we still had 21-fold variability. So you can imagine a patient is given a dose and the amount of drug that patient sees can vary 63-fold at peak concentration and 21-fold out through that kind of over total exposure, which is a lot. It's a lot of variability. Might that be contributing to why some patients respond and some patients don't? So the other thing that we were able to look at in this particular study was the impact of this variant. And so again, from our in vitro studies, it seemed to decrease the activity of this pathway, meaning that metabolism of naltrexone was reduced. And so in individuals who carry this variant, who had the dotted lines, we would expect them to have higher exposure to naltrexone because, again, their bodies are not able to potentially process it as quickly as those without it. And that's what we saw. So the individuals with the variant kind of clustered towards the top of this graph, suggesting that their exposure indeed was higher. So in addition to being highly variable from patient to patient, in addition to potentially having some impact of the genetic variant on exposure, we also looked at variability in the metabolite, 6-beta naltrexol. We know that naltrexone seems to be most important in treatment, but now naltrexol, the metabolite, is also active. It's less active than the parent, but it does retain some activity. And there have been some reports that it's associated with some of the side effects. And so we did want to look at that as well. That was not as variable from patient to patient, and we didn't see a clear impact of genotype on 6-beta naltrexol concentration. So looking at the impact of genotype, we also were able to kind of query another variable, if you will, the impact of food on naltrexone exposure. And because this cohort of 21 was made up of kind of two separate studies, one where naltrexone was administered in the fasted state where they hadn't eaten since midnight, for example, and the other one was in the fed state where they had eaten within the past hour. And so looking at those differences, on the left-hand side is area under the curve, that total systemic exposure. This first kind of grouping here, you have wild type and then the individuals who are carriers of that genetic variant, substantially different, right? But then also looking at comparing these two to the fed state, genotype seemed very clearly to be involved regardless, but fed state also seemed to have some impact in increasing exposure, more so in the area under the curve than in Cmax, but was of interest as well. And then I wanted to share this particular example of a patient who actually completed 3 different study days. And so we were able to kind of look at within individual variability. Here you have this individual completed 2 visits in the fasted state. It's the blue and the gray that kind of have, fairly similar kind of concentration time profiles if you were to kind of extend and connect those dots, if you will. And relative to the fed state, this individual's fed state exposure was quite a bit higher. And you can see that reflected in the ratios here on the left-hand side. So We think about, we've got two different studies, two different time points, one in the fed state, one in the fasted state. Food status seems to matter, genotype seems to matter, but what else might be important? Before we delved too far into that, we were wanting to look at the kind of effect of these two large, what appeared to be important variables through linear mixed effects modeling. And so here, the AKR1C4 wild type had a fairly large effect, such that individuals who were wild type had reduced exposure, which again is consistent with what we would expect. Those who have the variant have higher exposure, and so on. And then fed state also seemed to matter. And just these two variables alone accounted for about 82% of the variability in area under the curve. So we took kind of 21-fold variability from patient to patient, and we're able to explain 82% of it just with their genotype and whether or not they received drug in fed or fasted state. We always like to think about what other potential contributors are there to this variability. Again, we can explain 82% of it, but that's still 18% left to be explained. We ask ourselves about disease severity. When we have a drug that's administered orally, it has to get absorbed through the GI tract. When there are disease-specific changes to kind of GI health or motility, that could certainly impact absorption and exposure. And so that was one of the things that we looked into. We also looked at differences in concurrent medications from study 1 to study 2, and those were essentially the same, minimal changes from study 1 and study 2. So kind of thinking through this process, it didn't appear that there were other substantial variables that kind of jumped out to us that could impact the variability or account for the differences beyond what we focused on. So in summary of that kind of second part, systemic exposure in patients treated with naltrexone is highly variable. It's influenced by food and by this AKR1C4 genotype. And of course, we always have to consider other potential variables, such as disease severity, that could impact drug disposition. So we've done in vitro work, kind of looking at some things. We've looked at patients and charted the course of what systemic exposure looked like. But what does it all matter, right? How does exposure link to response? And that was really the next question: is there a target exposure that we need to reach to get to response? And is it worth trying to control for some of this variability to reduce that, to identify the right dose, to target a certain exposure? But in order to do that, we have to figure out how to define response, right? And in psychiatric conditions, it's a challenge. There aren't great biomarkers to detect kind of objective response. So pharmacodynamic biomarkers are essentially this tool. They can be an objective measure of drug response. And specifically, they can detect target engagement. That would be like a lock and key type mechanism. If you think about PET scans that look at a receptor displacement, that would be like that target engagement. You can look at pathway modulation. In our case, we were interested in like reward pathway modulation. So not as granular as the lock and key, that receptor binding, but perhaps a step back, looking at the entire pathway and modulation of that by drug. And then finally, you can also look at disease-related change. That would be something like a hemoglobin A1C for patients with diabetes, or blood pressure in patients with hypertension, right? Those are biomarkers of drug response, but they're also related to the disease. And kind of the promise of using these pharmacodynamic biomarkers in precision therapeutics are kind of this idea that you can take a whole motley crew of individuals, you can give a drug, and you can look to see whether or not response is detected. And if you have this group that does have response based on this biomarker, perhaps you can further stratify them. You can look at the relationship between response and drug exposure to, again, kind of identify that target exposure. You can also correlate clinical symptoms with that particular response post-drug. And if you have individuals who don't respond according to this biomarker, perhaps those individuals need to be offered alternative treatment earlier on rather than waiting to see if drug fails, and also potentially offered alternative study to look at, alterations in exposure, whether or not there's receptor variability and other elements there to explore. So the idea is that a pharmacodynamic biomarker can be a really important tool to kind of tease out some of these individual factors and get patients the right treatment faster. But in psychiatry, that's challenging. As I mentioned, we're looking at brain response, and so it's a bit harder to get to, right? There are some biomarkers that have been explored in psychiatry, but nothing has been formally qualified. And I'll say that the FDA has a formal process of biomarker qualification that is generally done in the course of kind of a drug development program. And to date, there are no formally qualified psychiatric biomarkers, pharmacodynamic biomarkers, but these are some that have been kind of explored in the literature. So receptor binding with PET, as we mentioned, task-based quantitative EEG has been explored for stimulant response in ADHD. Cortical inhibition and excitation by transmagnetic stimulation has been explored. Lactate-induced panic attacks, so kind of this subset of individuals with panic attacks that when you administer lactate, that leads to a panic attack, and that could potentially be a biomarker, among other things. But regardless of these available, limitations remain, particularly in pediatrics, right? So we can't really use PET scans, generally speaking, for drug studies in kids due to the radiolabeled compounds. And so we'd like to identify some additional tools to make this a bit more objective. So that led us to this next question, is can we use neuroimaging as a tool to detect reward pathway modulation in teens with eating disorders? I pulled this graph here from the FDA guidance for biomarker qualification. And I just wanted to highlight kind of two elements when we're thinking about how to go about exploring or evaluating a biomarker. Kind of two broad buckets that one has to think about would be this analytical validation. So that's essentially like the measurement, how accurate that measurement is. If you think about a thermometer, you want to make sure that your thermometer is reading the temperature that it truly is, that it is reliable and reproducible and is consistent with its measurement, and that it can read a temperature across a dynamic range that might be important for, measurement of climate, for example. And so when we think about applying kind of that logic to neuroimaging, we want to make sure that what we're measuring is truly accurate, that it's reproducible across different measurements, and that we understand the dynamic range of which it's able to detect difference. And then the other piece is clinical validation, which happens a little bit later on, where you're basically saying, okay, we expect this drug to cause this effect, and we're looking at reward pathway modulation in this case, which is biologically plausible based on what we know about the drug's mechanism of action. And then finally, to correlate with clinical outcomes such that, are we able to see changes in the brain following naltrexone that are associated with reduction in binge purge behaviors over time. So these are the questions, these are the ways we're thinking about this. And for this piece, we'll kind of focus on the relationship between plasma exposure and brain response. We can also ask ourselves some additional questions here, similar to that kind of checklist I showed early on, but everything in due time. So we started with a pilot study to really see if neuroimaging, and specifically functional MRI, to look at that pathway modulation, could detect a difference post-drug. Can we even see anything? And so we set out on an open label single day study. It's a pretty long day for these patients, but they were teens with eating disorders between the ages of 13 and 21. They came in and they received a baseline fMRI where they had two different tasks while in the scanner, one to probe kind of general reward. We used monetary incentive delay. And then we also did what we kind of considered a disease-specific reward task, which was a passive food view paradigm. And so they had that kind of fMRI, and then they had oral naltrexone following that baseline scan. And then they had an fMRI, the exact same paradigms, 2 hours post-drug. And we chose 2 hours because that coincided with what we would expect the peak concentration, that Cmax, to be based on some of our earlier studies. And then we collected blood and urine, and they did a variety of questionnaires and assessments and so forth. And so our questions with this study were really, again, can we detect any change in brain activation? And if so, because we're looking at functional MRI, we wanted to know what part of the brain, what part of that pathway seemed to have the most change. So what region of interest was most responsive to naltrexone, and then which task, whether it was the general reward, monetary incentive delay, or disease-specific passive food view, led to kind of the most modulation of this pathway. So these are the early questions, and that kind of falls within this top, kind of pre-analytical evaluation, I would say. And so what we were looking for on fMRI was, again, kind of the percent blood oxygenation level dependent change post naltrexone compared to pre. And this figure here is representative of a particular patient in this study, where the orange kind of area here, that's the nucleus accumbens, their percent bold at baseline, you can see it was 0.19. And then following naltrexone, there was a substantial reduction in bold activation with a pre-post of minus 0.4 in this particular example. So that's what we were looking at the individual level. So we didn't want to, although group analysis can be informative for this particular study, we wanted to look at the difference within each individual, which was our kind of primary focus. This box plot here on the right-hand side kind of shows those two different tasks that I mentioned, the monetary incentive delay on the left hand, passive food view on the right, and then two different regions of interest that we chose based on some prior work. And what we saw is that on the right hand side, this red box plot, following naltrexone, there was a reduction in activation of the nucleus accumbens, which is kind of that pure reward region, which is what we would expect. And following naltrexone, there was an increase in activation of the dorsal ACC, which is also what we would expect because it's thought that the dorsal ACC is involved in inhibitory control. So this would suggest that naltrexone might improve inhibitory control. So the ability to kind of, with the MID, with that reward anticipation, kind of be excited about a reward, but also kind of put the stops on, if you will, which would be important to prevent binge or purge behaviors or following a craving. And again, kind of putting this together, what we hope to do is to take this variable exposure, be able to explain some of it and figure out where we need to be, what is the target that gets us that response in the brain. This schematic on the right-hand side is truly that. It's an illustration for a grant, and it looks really nice and clean and lovely. And if only real data looked like that. But that gray part is kind of, again, that idealized kind of target exposure. This is what some real data look like from our pilot trial, where we were looking at the relationship between total exposure to naltrexone and that percent bold change in the ACC, that area involved in inhibitory control. And it seemed that there was a negative correlation, actually, interestingly enough, such that higher exposure led to kind of the opposite change that we would expect. So does this suggest that a lower range of target exposure is where we want to be? Something we'll continue to explore. And we're doing that in a randomized control trial that we launched about a year ago as part of my K. And this is now taking it the next step to from pre-post design, where a patient comes in and gets a baseline scan first, and then their post naltrexone scan. Now they're getting naltrexone and then a scan, and then placebo and a scan. And that's separated out into two different visits. There's a 2-week washout, and the order is randomized and double-blinded, such that the patient doesn't know what they're getting, study staff doesn't know what they're getting either. Both of those visits look exactly the same. We draw blood, we collect urine, they do a variety of measurements, and they get the same fMRI scan and paradigms. So that is actively enrolling, and we hope to kind of, it'll go over several years, but so far, knock on wood, so good. And our next steps are really to kind of look at that dynamic range. So we can look at the variety of exposure, we can look at how responsive the biomarker, right? So that percent bold change in the brain is following acute naltrexone. And even though these graphs here say doses, we always like to think about exposure because it's more informative from the individual level. We already showed that by giving, kind of equivalent of the same dose, you have this 21-fold exposure and 21-fold variability in exposure. Just dose alone is not that informative. So we can look ahead to the future to think about looking at a kind of a range of exposures and hoping to see kind of a correlative kind of range of response based on the biomarker. And the hope is that if validated through kind of additional study, that this can be a tool for drug development and drug repurposing. I don't anticipate or expect that clinicians who want to prescribe naltrexone will, send their patient to get an MRI in the clinic. But perhaps as we do some of this work in the research space, we can identify that target exposure. We can use model-informed drug dosing to take individual factors like genetic variation and provide recommendations about fed state and say, this is the dose that seems to be most appropriate for you to get you where we need to be to get that response. So I'm hopeful that, the case that variability in drug response may be related to variability in drug exposure, but in order to fully tease out kind of these relationships, we need better tools to define drug response, and pharmacodynamic biomarkers can help us do so by enabling kind of our identification of target exposure, precision use of existing drugs, so getting better at using the drugs we have, and then also developing more effective drugs in the drug development space. And, as we think about dose exposure response, when we have a problem that starts with kind of variable response in the clinic, you can walk backward to say, what is our exposure like? And then finally, what dose do we need to get back to the response we want? So I'd like to acknowledge a variety of folks. This type of work takes a whole team, a whole lot of folks, and I have a great team and great mentors that help this work. And with that, I'll take any questions.
I'll just go ahead and unmute since I'm hitting all of my emojis or whatever. Anyway, it was a wonderful talk. I really appreciate you doing this, first of all. But frankly, just an elegant series of studies going across multiple levels. It's fascinating work and frankly, a real model broadly for psychiatry and drug development beyond eating disorders. My question is really, I'm fascinated by the imaging piece and the data that you had for ACC. I think that was ACC, looked a little bit, you mentioned it was kind of flipped in terms of what you were expecting. And to be clear, I can't count the number of times that's happened in my own data. So that just happens. We're working with humans and the brain is, we want to predict a lot, but it's hard sometimes. And so, but my real question then is what does the animal model literature say about this? And so do you have good models that you can reference back to in thinking about how exposure may relate to neurophysiology and focal effects within either ACC or within accumbens, and how that's informing your work moving forward?
Yeah, that's a really great question, and I think a challenge with eating disorders specifically, because there's a lot of kind of complex processes likely happening and interacting that lead to behavior. And we're simply looking at changes in the brain kind of acutely post-drug. We don't know what the relationship is between those acute post-drug changes are and clinical response, but hope to be able to get to that at some point. But speaking to the animal models, I think there's a solid argument to focus on the cleanest possible kind of relationships, like taste, for example. If we think about like taste receptive or taste responsiveness versus our passive food view. When we think about passive food view, we have these other things that come into play, like visual processing, although we try to control for, and Laura can speak a ton about this much more eloquently than I can. But it's different than if we were to deliver an actual substance, right? Where we can look at brain-related changes following delivery, which would be much cleaner and have a stronger tie to the animal literature. So something we've thought about. It's a really good point, and I think we'll have to consider it as we write additional grants and look further.
Yeah, no, I mean, and just from this speaks to my naivete in this space. It's like, so we do like in the neurodevelopmental disorders research world, we have pretty good models for at least some of the conditions, specifically some of the more simple genetic conditions and monogenic conditions. So you have really good models that you can rely on there, but it's sounding like that's much tougher in the context of eating disorders than I would imagine it would be. So yeah.
For sure.
Stephanie, did you use just the traditional MID with the monetary reward, or did you use, I know that there are certain modified versions that people have developed, like there's one in autism that uses high interest electronic type items. But it sounds like you were kind of trying to get at just sort of what we traditionally think of as reward, and then the passive food viewing task is more specific to food reward.
Right. Absolutely.
Highly palatable food items, or I'm just not very familiar with that task.
Yeah. So the MID we chose because of the ABCD trial, that longitudinal adolescent brain development kind of cohort, and we kind of matched our parameters to what they were doing to the best of our ability. And so my hope is that over time, as we get more and more participants through and larger data, we can then potentially compare to like historical controls, right? Looking at normative MID response from ABCD. For the passive food view, I mean, ideally we would do like a food delivery, but we're not equipped for that at this point. And so Laura had validated, Dr. Martin had validated a set of images in another population looking at food and then animals. So they saw food and had a variety of like kind of ratings of there were high appetizing, there were low appetizing foods rated by a different group of individuals, not patients with eating disorders, which is something to consider. But and then animals. Interestingly, I didn't show this, but in our teens, naltrexone kind of reduced the kind of what we would expect reward activation to the animals, and also animals seem to be more rewarding than food.
So just a practical question with naltrexone, is that something that you would deliver, like is it something that a person would take daily, or is that something they would take when they feel like a desire coming on to binge eat? Or I'm curious how that works.
Yeah, it's a good question. And I don't know that the optimal dosing strategy is established for this particular kind of target behavior. Clinically, I think both are utilized. More commonly, a daily administration of naltrexone is recommended to reduce urges and then behaviors. The issue with like PRN use is if you have an urge, that medicine is going to take an hour or two to get into your system to reach Cmax, right? And then, you know, how good are your coping skills for that hour or two? Right. Although I have seen prescribers use it that way, I don't think it's most effective based on what we know about how the drug works. So, but the other piece is that once it is administered, it seems to kind of have occupancy at the receptor for at least 24 hours, potentially 48, which is one of the reasons we created such a long washout period, to make sure that individuals who received naltrexone, there wasn't any kind of carryover effect that would be occurring if they received placebo second.
Got it. Thanks.
There is injectable naltrexone too, and we can talk about that if anyone's interested. But
do people have any other questions for Dr. Stancil?
So when are we going to build this device, Stephanie?
The food delivery?
I was going to call on Bill, actually, so.
Yeah.
Say it again?
I was going to call on you if you didn't speak up anyway.
Yeah, well, I read your mind.
Laura and I are working on it.
Okay, because
we'll probably put in an equipment, a School of Medicine equipment grant, because there's other studies as well as some of our researchers researching alcohol use that could potentially, although that would be a whole nother set of hoops for us to jump through.
That's what we're here for. Get Manish working on it.
There you go. Sounds like you have an invitation then. So
Yes. And lots of good work ahead, hopefully.
excellent. Well, I want to thank you again then for doing this, Stephanie. This is like I said, just a really fascinating line of work. I hope to hear updates on it as the imaging data starts rolling in too. But like I said before, I think it's just a really strong model that's probably much broader than eating disorders. It is much broader than eating disorders. So I appreciate you sharing that with us, and thanks for the wonderful talk.
Thank you. And thank you all for the invitation. It was great. I appreciate it.
Absolutely.